Good day to all of you. I'm Vanati Perumal, the upcoming CEO of small CTC diagnostic company based in Perth. Thank you, Hanson, and the organizers for the invitation. I'm excited to present my work at this meeting with the industry professionals. My disclosures are Sir Charles Gardner Hospital Radiation Oncology Fellowship, Curtin University Med School Research Funding, and Australian Technology Network University Sydney Commercialization Training Workshop Travel Grant. I'm here to share my pilot clinical trial experience with Curtin and Charles Gardner Hospital during my radiation oncology fellowship between 2017 to 2019 for new gastric cancer drug discoveries and development platforms for anti-cancer resistance and sensitization mechanisms. I'll give you an overview of human predictive circulating cancer stem cell fear blood testing to accelerate Wnt drug discovery and development process outside the patient for precise treatment selection with less complications and toxicity. We are based in Perth as a small business startup cancer diagnostic company with proof of concept, CTC, and circulating cancer stem cell sphere, cancer stem cells, and SFRP4 gene therapy expertise. I will also discuss how SFRP domains, small molecules, can be genetically tested, modified for therapy resistance and sensitization mechanisms for Wnt-dependent cancer risk patients. It will help our SFRP4 and cancer stem researchers to achieve their therapeutic clinical goals. So these are my publications and manuscripts during the funding period 2017 to 2019. My talk comprises of 4 components: introduction, highlights, and conclusion and future directions. An introduction, I will talk about gastric cancer incidence and prevalence. And in highlights, I will discuss about how do we simplify gastric cancer drug development platforms. And in conclusions, I will discuss about new technologies intervention. And in the future directions, I will discuss what I'm aiming to do. So every year, around 990,000 people are diagnosed with gastric cancer worldwide, of whom approximately 738,000 die. And gastric cancer is the fourth common incident cancer and the second most common cause of the cancer death. And the risk factors are family history, diet, alcohol consumption, smoking, Helicobacter pylori, and Epstein-Barr virus and EBV infections. And countries lacking the screening program are detected at the advanced stage. In most Western countries, the 5-year survival rate is less than 20%. And survival rate can be improved if the cancers are diagnosed at the early stage with the appropriate screening program, particularly where we cannot access or the cancer is in a risky location, avoiding the complicated invasive procedures. Finding also suggests that malnourished people are prone to gastric cancer, and tissue biopsies CSC does not reflect current cancer genomic signature status. The cancer keeps changing with treatments. To help doctors understand the cancer better before symptoms appear or even tumor mass forms, that is, before patient radiographic imaging progression. ctDNA and the cell-free DNA detections are not at the cellular level and are after risky internal drug treatments, exposing to drug toxicity, side effects, and unwanted cost. To tackle recurrence and reducing more invasive and painful procedures on different spot biopsies, most importantly, to understand the whole body tumor heterogeneity for effective treatments, we need to introduce simple blood testing to assist the drug development selection process faster with more accuracy and sensitivity. These are my PhD and part-time research publications credentials from Curtin University. with full funded Curtin International Research Scholarship and stipend through national and international collaborations. Previous studies have shown secreted frizzle-related protein and in particular SFRP4 are downregulated in mesothelioma tissue and cell lines. In addition, we also demonstrated promoter methylation of SFRP4. Our laboratory has previously shown downregulation of SFRP4 in mesothelioma cell lines. We investigated vesicular trafficking of SFRP4 and observed that the natural like domain has a vesicular association signal. More significantly, intracellular redistribution of SFRP4 was observed in response to Win3A, suggesting Win3A can modulate intracellular localization and secretion of SFRP4. In continuation to my PhD finding, we collaborated with Oxford University to study the function of SFRP4 domains and mutant proteins on cancer stem cells. As shown here, the GBM cancer stem cells showed significant effect on NLD domain treatment, and these are unpublished data. As shown in the figures, we further examined CD133 and LDH expression in malignant mesothelioma stem cells. We continued looking at the effect of SFRP4 and its peptide alone and in combination with cisplatin and reduced cell proliferation and disrupted the tumor sphere formation. For the first time, we identified that SFRP4 and associated peptides could help to sensitize and destroy tumor-initiating stem cells from two mesothelioma cell lines, which may lead to the development of an effective treatment to combat malignant mesothelioma. In the future, in continuation to my CTC study as a new therapeutics, we will examine the effect of our peptides using human CTC spheres therapeutics with the view of implementing their use in the clinical blood testing setting. Coming to gastric cancer background, these are the patents and publications, available literatures. So Peter MacCallum Cancer Institute published on measured gastric cancer patient blood and tissue SFRP4 biomarkers. And the New South Wales group established long-term EPCAM positive and negative CTCs on metastatic cancers. And another group, a Korean group, they published on gastric cancer stem cell using patient tissue. So in bridging the gap for unmet clinical need and as a novel SFRP4 therapeutic target for gastric cancer based on circulating cancer stem cell is not studied and established. In improving the available testing with family history, rare pathologic variants, and single nucleotide polymorphism SFPs, CTC germline and somatic mutations will be sensible addition to cancer risk and prenatal setting population to identify the cancer risk. We could also monitor CTC and CSC therapy resistance variant or rare pathologic variants. SFRP biomarker SFR expression is elevated in blood samples and tissue on different stages. So until now this is known, but we have not explored the role of SFRP4 on CTCs in gastric cancer. So where are we in CTC discovery? So what we know and what are the areas explored and what is the commercial scope for new circulating cancer stem cell sphere drug screening, and what is the unmet clinical need and what improvement is required. And here I will discuss my own findings. As incidental finding, Viable CTC line and tumor spheres were cultured in cancer risk patients. And established single-step viable head and neck cancer CTC and circulating cancer stem cell sphere count for radiotherapy predictions. In translating our publications as a new blood product therapeutics, CTC and CSC spheres were tested for SFRP4 and cisplatin. So our mission is same as Vortex microchip intact live cell technologies, introducing live CTC technologies for understanding the CTC biology and drug development will have huge commercial potential. Our technology could be automated with and scaled up to 50 mLs for patient circulating cancer cell drug development. And to support microfluidic device size and scale up volume limitations, we need to improve technologies to capture all types of CTC intact in larger volumes for patient-specific cell drug manipulation and delivery. And this technology could also apply to other disease model settings for genetic testing and prenatal setting blood testing stem cells therapies. And what role does SFRP4 CSE therapy play in radiotherapy, radiographic imaging, chemotherapy, and other anti-cancer therapies, and early detections, including coronavirus cancer patients for monitoring resistance and sensitivity. So, what the new generation CTC technology could offer? So we claim as all-in-one compact capability product for label-free head and neck cancer CTC count and viable head and neck cancer circulating cancer stem cell sphere count and the first viable cancer risk CTC line count and the first viable cancer risk SFRP4 circulating cancer stem cell sphere dissociation. And we simplified as a single step with faster turnaround time. So commercially available technology provides the combination of dead and live cancer cells mostly focused on metastatic cancer. So we need to improve to the existing CTC technologies models. So as an inventor in bridging the gap of CTC and SFRP4 drug development, developed my proof of concept products from head and neck cancer and from a radiotherapy feasibility study in compliance with NHMRC and WA Health infrastructure. And trial contract is paused and discontinued due to funding issues. And in pandemic with hopes, started my own small business company. The methods are embarked for trial completion and patent development. And these are pilot trial products. And the products are viable head and neck cancer CTC count with high recovery, specificity, and purity for radiotherapy predictions. And viable head and neck cancer circulating cancer stem cell sphere count with high purity, that is about 100% in 18 hours, faster turnaround time. And fourthly, the viable CTC line and sphere culturing for early cancer detection as my incidental finding. And further to our previous mesothelioma cancer stem cell peptides finding, introduced viable CTC, SFRP4 and cisplatin combination treatment as an improvement to previous tissue cancer stem cells publications and expired patent products. Interesting cancer risk cluster dissociation effect was noted with limited cultured cells similar to Cell Paper Swiss group on FDA duct screening using mouse model circulating stem cells. So I'm happy seeing the effect of CTC cancer risk CTC cluster dissociation with SFRP4. So still there's a scope for other solid tumor detections early cancer screening and prevention with more than 150 billion USD cancer stem cell cancer diagnostic market need. I will share some interesting finding for future collaborations to bring quality product to the market as high standard labs for high impact factor publications data. We used appropriate positive control and negative experimental controls for organ specific cell lines with statistical significance and longitudinal follow up studies. Here we used three spiked head and neck cancer cell line for measuring patient metastatic sphere culturing potential with 98% recovery, sensitivity and purity on spiked head and neck cancer cells for radiotherapy treatment. And these are live CTCs and the CTC lines. As shown here, these are live circulating cancer stem cells we are culturing. As a single step faster turnaround technology, our lab-based test allowed to quantify the number of viable tumor cells in the blood in 18 hours turnaround time. As shown here, these are single step CTC spheres isolation, and these are positive early cancer circulating cancer stem cell spheres and late cancer stem cells here with site population, and the negative control did not show any sphere formation, and the bottom panel shows the conventional density gradient with side population spheres, and the methods were also simplified to multi-step CTC enrichment, and we were able to cryopreserve, revive and viable solid tumor morphology cancer-associated fibroblasts like CTC lines. Additionally, we were able to generate numerous primary and secondary spheres from the same wells, which is interesting. As shown in the figures, these data are from healthy control early cancer risk CTC line testing. Surprisingly, during validation, the 5 volunteers out of 13 negative control groups yielded 6 to 10 days viable CTC lines, and these lines were revived on cryopreservation, and the CTC line morphology were like cancer associated fibroblast. The proof of concept study was on 22 to 48 years old healthy volunteers group, detected the viable CTC lines as 38.5% for risk and 61.5% free from cancer. In supporting this finding, a recent early cancer detection group published on stem cells reviews and reports on very small embryonic stem cells and OCT4 expressions with good statistical significance. And this group was from India and Singapore, and this was measured in an Indian cohort group. And our products are unique to CancerSeek and GRIAL, and with drug testing solutions and live cell immunostaining and genomics compared to known cancer tissue and organ signals, measuring the tumorigenicity and appropriate animal models for identifying cancer type will open doors to new interesting early cancer detection and prevention drug therapeutics. Required more organ cell type or inflammatory marker signals experiments, confirmatory experiments, and measures for early cancer screening and follow-up for cancer risk patients that's required. Real, false, positive and negative control percentage should be taken into consideration, and a decent follow-up study should be carried ahead to save these cancer risk patients in saving lives. Interesting incidental finding noted for future research direction. So, where are we now? To offer a solution to current treatment challenges, introduced SFRP treatment in short term CTC for complete elimination of cancer stem cell and cancer relapse approaches. New dose and cell model innovation is introduced, unique to my own previous published SFRP4 concentration on equivalent spheres with limited CTCs, and successfully generated numerous primary and secondary tumor spheres from spiked CTC lines for SFRP4 Wnt targeted preventive drug screening, alternatives, sensitization, and anti cancer therapy follow up approaches. As expected, SFRP4 dissociated the cancer risk aggregates. Oh, we established viable cancer risk 10-day CTC lines, 6 months tumor spheres and clusters. And we also established viable head and neck cancer CTC and tumor sphere counts for radiotherapy predictions. And interestingly, SFRP4 dissociated cancer risk CTC line clusters as shown in the figures. And SFRP4 with cisplatin showed a synergistic effect by decreasing cancer risk CTC line spheres. The feasibility study allowed us to provide custom-made circulating cancer stem cell blood testing services to build better patient representative drug testing for empowering cancer research and cancer care from head and neck cancer cell lines and healthy volunteers. The outcome of my talk and with global initiative will set the stage for external investor funding and industry partnership opportunities for advanced cutting edge technologies. So, as a measure, I wish to introduce routine live CTC blood testing and treatments for WA community as an occupational health hazard measure for asbestos-induced mesothelioma and head and neck cancer, breast, gastric cancer, and solid tumors through collaborations. Our early cancer detection incidental finding with further genotyping and immunotyping will open doors for cancer preventions. To advance cancer treatments, I'm open to large CTC clinical trials and circulating cancer stem cell sphere genomics collaboration for drug discovery and anti-cancer therapy follow-up approaches. So lastly, we required funding support from government, private, philanthropists for cancer screening and prevention programs from developed and developing nations for WA, India, USA, UK research collaborations on cancer risk population families and healthcare providers, especially to avoid detecting cancer at an advanced stage. And thank you for your time. This is my CTC team, Sir Charles Gardner Hospital and Curtin University team. I wish to thank Dr. Joshua Das and the team for the commercial project and mindset training, mentorship, grant writing, funding, and clinical trial opportunities. I wish to thank Sir Charles Gardner Hospital, Curtin University, and Australian Government Immigration for supporting international research innovation opportunities. I also wish to thank my mentor, Professor Arvind Dharmarajan, for mentorship and international grant writing opportunities. I wish to thank Archduchs Merck and Hanson Wade for grant success and opportunities and invitations. And these are my credentials open to collaborations. Thank you.